Analytics web. We will be recording this webinar, as you've probably all heard through Zoom. So be mindful of that. But we welcome everybody. Thank you for joining this webinar with Dr. Faranak Farzan's lab. Who will be presenting a little bit later on their exciting work through the Center for Analytics on an integrated multimodal biotyping toolbox for personalized health. Before we get to that, I want to first introduce myself. I'm Francis Jeanson. I'm the head of the Center for Analytics at the Ontario Brain Institute. And I want to acknowledge this land, its nature and its people, in particular the Anishinaabe, the Mississaugas of the Credit, the Wendat, the Haudenosaunee, and every other nations, recorded or unrecorded, who have taken care of this land long before us, land which we must continue to take care of together. So we'll start with a bit of background on the Ontario Brain Institute and the Center for Analytics, and then we'll jump right into the presentation from the lab members of Dr. Faranak's group, in particular from Dr. Chloe Stangal and Raj Chatterjee, who are part of the lab and will be presenting just after my background on the Center for Analytics. At the end of their presentation, we'll then take questions and answers. So that should be in the last sort of 20 minutes of our webinar today. So let me jump into a little bit of background about OBI and the CFA. So the Ontario Brain Institute is really aiming at accelerating discovery and innovation to really benefit patients and the economy. We have a very collaborative approach for research where we aim to bring together expertise across the neurosciences and the brain disorder areas that span multiple institutions and multiple organizations to create collaborative groups. And we call these integrated discovery programs, which are a cornerstone of OBI's vision and mission to help accelerate a better understanding of brain disorders and develop better ways of preventing them, understanding them, and treating them. We have also a very big component around commercial innovation, where we help bring the research and commercial innovators together to develop new technologies that can have an impact on patient health with brain disorders. And finally, we're really aiming at connecting that care with the patients and the populations of patients with these brain disorders. And so we have quite a bit of engagement through patient advisory committees and working with clinics and diverse populations across Ontario, but even Canada and beyond. One very unique aspect of what OBI has done was really around bringing together these institutions, as I mentioned earlier. And we formed these integrated discovery programs, such as CAMBIND, of which Dr. Faranak Farzan is a part of. CAMBIND focuses on major depressive disorder in particular, and is connected to all these institutions that you see in this network on the top left in red. We also have a program in neurodevelopmental disorders called POND, another program in cerebral palsy called CPNet, a newer program called CALM looking at multiple mental health disorders in children and youth, and finally, a major program in concussion called Transcendent that connects all these groups that you see here on the graph. So we have over 40 institutions that are connected, multiple users who are essentially collaborating and working together. And all of these research initiatives are collecting rich data that are standardized and that we combine and aggregate onto a central platform called BrainCode. So BrainCode is really OBI's neuroinformatics data platform that enables that standardization, collaboration, data federation, but also linking of data and ultimately analysis and discovery of these data. So this creates a really big neuroscience discovery opportunity. This diagram illustrates a little bit of this approach around looking at these major brain disorders in these columns, these purple columns that span across major brain disorder areas like cerebral palsy, concussion, neurodevelopmental disorders, mental health, depression, and others. But as you can see, we need to collect data across a multitude of modalities. That includes clinical, neuroimaging, genomics, molecular, wearables, and sensors, and many more. This is what you see in these blue rows. And so this really spans across these major brain disorder areas so that we can essentially start understanding these brain conditions in a much more fulsome, more fulsome way, but derive and understand the complexities of these disorders much more clearly. So this has created the opportunity for us to not only enable these groups to collaborate, but also make these data available for sharing and secondary use. So we have this rich multimodal data on the platform. We enable that data sharing. We have common data elements that help standardize what we collect. And ultimately, we can also help linking that data with other repositories and other databases, which enables these researchers to then explore the brain disorders in much more depth. And so far on our platform, on the BrainCode platform, which you can go and visit, it's braincode.ca, so you can see the link at the bottom. We've made a number of data releases available for researchers to explore, from the Pond, CPNet, EPLink, CanBind, Connect, and Andrew program, just to name a few. And there's quite a few more if you visit the website. However, we've noticed over the last few years that there are still challenges in this area of innovation in what we call neuroanalytics. There's still a gap related to the number of skill sets that we have available that can look at these complex multimodal data and derive insights from them. We also need to help the labs and the startups who are developing new ideas to accelerate those ideas. How can they leverage data? How can they leverage modern computing, especially for machine learning, like GPUs and larger compute clusters? And we also need to help them think about translating. How do they bring these ideas to the bedside so we can have impact on patient care? And these are complex challenges that are not easy to navigate. And so what we've done is create the Center for Analytics to precisely help labs and startups navigate these challenges. So those three pillars are really at the core of what we do with the Center for Analytics. So developing skill sets, through access to resources, through also sharing of training materials, and ultimately creating a community of practice around how do we work with these multimodal data and develop new models. We also help with the acceleration of these labs and startups through access to high quality data, access to advanced computing, and a growing ecosystem of tooling in this space. And finally, on the translational side, we're providing additional support to help with the validation, for example, of some of these analytics and tools that come out from these projects. We can also help navigate the regulatory pathway and privacy requirements by providing access to secure computing, but also helping them navigate the challenges around regulatory approvals, for example, when it comes to bringing a solution to a clinical context. And ultimately, we can help also in the space of advancing the productization of apps. You may have something that's in the lab in a preliminary form, but now you need to make it available for a broader audience. How do you productize that so that others can make use of it in a way that's robust, reliable, and also accurate? And so what we've done is really launch a number of Center for Analytics initiatives, as we call them. And we're currently in our second cohort, of which Dr. Farzan is a part of with her lab. And we've created a webinar series essentially to help illustrate the exciting work that are being done through these initiatives. We've had so far two webinars over the course of the summer from AimSense as well as Netramark. Today is our chance to hear from Dr. Farzan's lab, and we will have future webinars coming up in the next few months. So stay tuned for that. We're also going to be announcing our third cohort later in the year or early next year. So stay tuned for that as well. Before I pass it on to Dr. Farzan's lab, I just want to share a few resources that everybody in attendance here can take a snapshot of or at least record. We have methods tutorials that are coming out from these CFA initiatives that are posted on our GitHub. We currently have two from the first two projects. The current 8 projects will be contributing to these methods tutorials in coming months. And ultimately, the webinars that we have, including this one, are recorded and listed on our YouTube channel, which you can link to here. So thank you for this little bit of background on the CFA. And I'll now move to passing it on to Dr. Farzan's lab, in particular, Dr. Chloe Stengel and Raj Chatterjee. So Dr. Chloe Stengel is a postdoctoral fellow at the eBrain lab led by Dr. Farzan. She received her PhD at the Paris Brain Institute, studying the modulation of cortical oscillation and brain temporal complexity by non-invasive brain stimulation for the improvement of visual attention. Dr. Stangel is currently pursuing her interest in the functional role of brain complexity by investigating brain complexity as a biomarker of antidepressant treatment response and its potential for biotyping depression patients. Then we will have Raj Chatterjee, who's a PhD candidate at the SFU eBrain Lab as well. He has a background in mechatronic systems engineering from SFU with industry experience in systems design and product engineering and training in neuroscience and signal processing. Raj has developed several toolboxes for data preprocessing and analysis for multimodal brain stimulation and recording research. He has most recently developed the integrated multimodal biotyping, or IM biotype Python library and toolbox alongside the eBrain Lab team to share with the OBI community. So thank you both Chloe and Raj for joining today. I'll pass it on to you, Chloe.
Thank you very much, Francis, for the introduction, and thank you for having us today. We're very excited to be here to present to you the development of our toolbox that we're doing. This project has been made possible by the funding we received from the Center for Analytics, so we're very grateful for that, and thank you for the great introduction. So what we'll present to you today is our toolbox, which we call I'm Biotype, or an integrated multimodal biotyping toolbox for yielding personalized health. So I'm Chloe Stengel, as Francis introduced. We also have Dr. Faranak Farzan, the lead of our team, and also part of the project, but he will not be presenting today, but he's present for this webinar, is also Dr. Paul Dehami, and he's an expert in using biotyping in the field of depression, and I'll also present some of the work he's been doing, and he's been very helpful for this toolbox as well. So first of all, I'd like to introduce what we're trying to address with our toolbox. So in psychiatry today, the common approaches is that patients are given a categorical diagnosis. So it could be depression, could be schizophrenia. I'm sure you know other diagnoses. And then they're prescribed a treatment for their ailment based on this diagnosis. And this approach assumes that every single patient will respond the same way to the treatment, meaning that they have the same symptoms that are going to be improved the same way by the treatment, and the treatment will work the same way in every patient. But that's not actually what we observed in practice, in clinical practice. What we see is actually low response rate for first-line treatments. So those are the treatments that are prescribed first after the patient gets their diagnosis. And we can see up to 30 to 50% of patients who don't respond to treatment. That's obviously a huge issue in the field of psychiatry because it contributes to the burden of disease for these patients if we're not able to find very fast a treatment that's effective for them. So how do we deal with this? So in reality, neuropsychiatric illnesses are highly heterogeneous. So people that are grouped together, that are given the same diagnosis, can actually have very different clinical and biological profiles. And what we mean by this is that they can express different set of symptoms, and they have different processes that are altered in their brain because of their disorder. And so this heterogeneity leads to differences in treatment response and in illness evolution for every single one of the patients. Our solution to this is try to better understand the underpinnings of their disorder. That means understanding the biology. That could be using varied data modalities, such as genetic marker, neuroimaging, so recording of brain structure or brain activity, or measures of behavior, and then also looking at individual symptom expressions for these patients. And then based on this, we can cluster the patients, not based on their diagnosis, but try to find groups of patients that have similar biological and clinical profiles. And then we can prescribe a treatment specific to the cluster the patient is in. And so this can allow better understanding of the disorders and support better treatment decision. And so this can lead to more effective and personalized treatments for these patients. And this entire approach is called biotyping. And so biotyping describes this general approach to group patients with a similar biological and clinical profile to support then treatment decisions. So this is all well and good. And biotyping is very promising, and there's been more and more papers using this approach over the last 5-10 years. But in practice, it's actually quite hard to achieve. And this is because of a few challenges. One of them is the challenge of replication. So here I've put on the slides a few papers that I've published only within the last 5 years that highlight this issue. And so what we mean by this challenge of replication is that a research team will have data recorded from a set of patients. They'll find different biotypes that they think are clinically relevant and can really help for clinical decision for these patients. But then a different research team looking at the same diagnosis or groups of patients will not be able to find the same biotypes again. So this is really a big issue because then it addresses the robustness of the results that we found. And one of the reasons why this happened is that we need larger and better control studies. But also, this is not the only reason why this replication is challenging. And one of the other challenges that plays into this is that there is a huge diversity of methodological options for doing biotyping, for doing a whole biotyping pipeline. So here what I show on the slides are like the two most important steps of the biotyping. So there's data fusion. That's where we try to understand the clinical biological profile of each patient, and then the clustering that I've shown earlier. And you can see that for these two steps, there's a lot of methods available. And this doesn't only even list all the methods that we have available. For data fusions, you could do model-driven, data-driven, even within data-driven, a lot of different methods have been used. For clustering, it's the same. There's hard, soft clustering, a lot of subdivisions within that and different methods. And so for research that's coming in and wanting to do biotyping for a certain group of patients, it's very hard to know which methods to use, which methods are the best, the most robust, and will give us the best results. And also the fact that there's all these methods available is also made even more challenging to manage by the fact that right now there's a very fragmented landscape of software packages available to do biotyping. And there's not a single toolbox or software package available today that allows to do end-to-end workflow. So what we mean by this is that there are several steps within biotyping, which I've already addressed, and there's not a single toolbox that will do all the steps within the same structure. So here I've illustrated some of the most common toolboxes, software packages available today in different languages. And we can see like we have the different steps of the biotyping, data fusion, clustering. Some of the toolbox will do data fusions, but not clustering. Some will do the clustering, but not the data fusion step. And none of the toolboxes have a graphical interface, meaning that for a lot of them, it's kind of challenging to get to know and to use these toolboxes. It's more for expert users. It's not very intuitive to use. So our solution to all of these challenges is the toolbox that we're presenting to you today, the AmBiotype toolbox. And when we developed this toolbox, we really wanted to address each and every one of the challenges that I presented. So for the challenge of replication, we make sure to have a biotyping methodology engineered for robustness and multimodality. Linked to this, to also address the challenge of choosing the appropriate methodology, what we've done is we've taken some validated workflow, implemented it in a toolbox, and then we've also implemented other ways, such as hyperparameter optimization, so that the toolbox can find the best parameters to use on your data. And lastly, for the third challenge of insufficient options and no end-to-end workflow, our toolbox is a complete Python-based solution, doing all the different steps of biotyping as illustrated right after. And we also have a graphical interface so that it's easy for new users to use. We really want this toolbox to be very intuitive to use. And we also have a scripting library and scripting language for the more expert users. And so this project, the AmBiotype Toolbox, is really designed to promote standardization and best practices for the growing field of biotyping in psychiatry and beyond. Our toolbox is based on a very simple but powerful architecture with these four main steps. There's a data interface where we choose the data we want to come into the toolbox to do the biotyping. There's a data fusion clustering step that I've already gone over. And then we have an evaluation step, or we'll give the user metrics to know how good and robust the results are. So to go into a bit more detail here. So for the data interface, the users can input their raw data in a CSV format. There's a few basic data processing functions that are available within the toolbox. And then from this raw data, the user can choose individual feature vectors. So these features vectors are basically different measures we have from the brain, from the clinical scores, from the behavior that are going to be used to understand the clinical and biological profile of the patients. So these individual features then go into the data fusion step. Before data fusion, we also have some feature preprocessing steps directly available within the toolbox. Those are all optional and they're illustrated here in the bottom. So there's a lot of different steps you can do. Some of them are, for instance, you can take out some variants from the data from variables that you're not interested in. There's options to do standardization, normalization of the data. And there's also several options to do what we call dimensionality reduction. So dimensionality reduction means that we kind of try to reduce as much as possible the features that we're going to use to go into the data fusion clustering. And why this is important? This is very important for the replicability and the robustness of the results, because it's very much known that when you do this kind of analysis, if you have a lot of features but not enough subjects that you record from, this can lead to things like overfitting, or it can in general lead to a solution that's not going to replicate in another data set. So we want to avoid that. So we have these options available for dimensionality reduction within the feature preprocessing or within the toolbox. You don't need to take your data anywhere else to do this. So once we've done this step of feature preprocessing, we go into the data fusion. The method we've implemented to do data fusion is canonical correlation analysis, or CCA, and we also have a few variants of this method. And right in the next slide, I'll explain to you what this method does. But basically, just to say it very quickly here, it kind of fuses data from different modalities. So for example, brain imaging, clinical scores, fuse this data together. And so at the end of this, we have what we call data fuse features, and those can go into the clustering step. You can also use directly the raw feature vectors from the data to go into the clustering step with the feature preprocessing, again, that you can do optionally here. So in the clustering, again, grouping of our patients into different groups. This is done with different methods implemented, k-means clustering, hierarchical clustering. I'll explain very quickly in a few slides what this does. And at the end, we have the visualization of the results, and we have some metrics to evaluate the robustness and reliability of the results. So that's really the architecture of the toolbox and what it does. So as promised, now I'll go a bit more detail into the data fusion step, and especially the method of canonical correlation analysis, or CCA. So what this method does is it identifies the sources of common variation in two data sets. So here we have data set 1, which is data X, data Y, and they're both in two different domains, domain 1 and domain 2. What we mean by this is that there are different modalities. So for instance, domain 1 could be brain imaging data. So we measure the activity of your brain. And so this is, you have one measure for every single participant that you record from. And then in the columns are what we call variables, sometimes also called features. So this could be one measure of this brain activity. So variable 1, for instance, could be measure of brain activity in one brain region. And then variable 2 could be measure of brain activity in a different region. So we take all of this data together into domain 1, and then we have domain 2, completely different modality. So this could be, for instance, clinical scores. So again, we have all the subjects that we record from, and the different variables this time could be, for instance, evaluation of a single symptom for the disorders that is several symptoms together. This domain 2 could also be behavioral measures. So here I'm illustrating some examples, but really what to understand about the CCA method and what is so powerful is that it's very flexible. The domain 1 and domain 2 data could be anything you want. So then once we have these two data sets, CCA seeks to maximize the linear correlation between the two sets of variables that we have here. So to get a bit technical just for a few seconds, so the way this does that, it does that by finding a linear combination of the different variables in each data set. So that's illustrated here. And then this linear combination of variables in both domain 1 and domain 2 are done in a way that they maximally correlate. So this is what is illustrated here with domain 1, domain 2, and this canonical correlation, this strong correlation between the two domains. And this is what we call the mode. So the mode is what kind of fuses the data from these two data sets together. So really the big picture here of the CCA method is that it can find combinations of biological variables that are related to combinations of specific symptoms or related to combination of specific behaviors. That's how the two data sets are fused together. So this is a multimodal method. And what's really important about multimodality and looking at two different modalities of data together instead of looking at them separately is that you have a more complete overview of the profile of the patient when you fuse these modalities together. And so if we study these modalities separately, we might miss some information that's in there and that's important. So that was the data fusion step. Now moving on to the clustering step. So this is where we separate the different patients we have recorded into different groups. This is done by a variety of different methods. Some of the most common methods here are k-means clustering and hierarchical clustering. I'll explain the k-means clustering. So what this does is here we have in blue all the data points. So this all of our subjects recorded, for instance. And it will try to draw clusters within this data. And what a cluster means is that patients within a cluster are closer together than they are from any other patient in a different cluster. So the way this is done is that we'll define a centroid, measure the distance from every data point to this centroid, find the closest one. So that's done by Euclidean distance. This will be iterated several times by slightly moving the centroid each time until it converges. And you have these two clusters, like the red cluster, the green cluster. And so, as I've said already, all the patients within the green cluster, for instance, are closer together than they are to any patient in the red cluster. That's one way of doing the clustering. There's a lot of different ways of doing this, as I've already illustrated. So very quickly, this is hierarchical clustering and especially the agglomerative method. So in this, it's done slightly differently. So again, in blue are the data points. At first, each data point is considered its own little cluster. And then each step, like these data points and these clusters are brought together 2 by 2. So you have this kind of dendrogram representation. But again, here, this also represented the Euclidean distance between the data points. And the further away 2 data points are, the less likely it is that it'll be in the same cluster. So that's for clustering. And so here I've really illustrated for you through data fusion's clustering, the whole biotyping pipelines and the methods we're using. And now what we want to do, once we have this pipeline, this workflow, we want to really validate it that it works and it gives us reliable and robust results. So here I'll present to you a study done by Dr. Paul Dehamy, which is currently under review, to illustrate really what kind of results we can get with biotyping and how robust our pipeline is. So for this first analysis, we used a first modality of data. We used data from EEG, so electroencephalography, which is a measure of brain activity through time. And especially in this analysis, we're looking at EEG power. So EEG is comprised of different frequency bands composing the EEG signal, and we're looking at the power of different bands within this. So we know there is a link between EEG power and depression, but findings here are inconsistent. So to do this analysis, we used a big data set, which is the Kinepine data set, part of OBI, that Francis introduced a bit earlier. So this is a big data set of patients with depression. And we also aggregated other data to test the robustness of the results. I'll go to the next slide. And so just looking at the link between EEG power in different bands and a depressive symptom score. And what's presented on this slide is the results from the CCA step. So you might recognize a little bit what I presented earlier about this correlation between the two domains. So what we found in this analysis is there's one significant correlation between EEG power, specifically in the delta band, so that's low frequency, and depressive symptom scores expressed at baseline in the patients. So there seems to be some kind of relationship between delta power and depressive symptom score. Once we have this significant correlation, we want to understand which symptoms and which kind of variables in the delta power measure contribute to this significant correlation, this kind of mode of covariation between the two domains. So that's what's illustrated in figure B and C. We're looking at which symptoms contribute the most to this relationship. And what we found is that atypical symptoms for depression are the ones that contribute the most. So atypical symptoms include hypersomnia, include sleep symptoms, and other symptoms such as suicidal ideation and psychomotor slowing. And so it's the expression of these symptoms that contributes to this relationship that we found. And if we look at the distribution, like which kind of measure we have contributes the most to this relationship, we're looking here at what we call a topography. So that's the spread of activity over the head. So this is the head of participants seen from above. And we've measured this delta power at different sensors all over the head. And we're looking at like which sensors contribute the most to this relationship. And we found this decrease in delta power over occipital and parietal areas and over frontal areas. And that's the topography that we see. So that's the result for our data fusion step. As I've illustrated before, one of the challenges in the literature right now is that we're never sure if these results will replicate. So to make sure that our results are robust and will replicate in another data set, we've done external validation. So we've applied the same kind of CCA model to one data set, and then looked at if we take a completely external data set, completely unseen, do we see the same significant correlation between the delta power score and the depressive symptom score? And we indeed do. We see the significant correlation again. And when we look at which symptoms contribute the most to this relationship, we see again the atypical symptoms for depression, hypersomnia, sleep symptoms, suicidal ideation, psychomotor slowing. And we also see the same pattern of distribution of this decrease in delta power over the topography, so over the head of the participant. So we're confident that our pipeline that we've used here is robust and can produce reproducible results. So that was the data fusion. Now we're moving on to the second step of a biotyping pipeline, which is the clustering. Using k-means clustering, we've identified 2 different clusters in the data. So that's, again, the spread of the different patients over these 2 domains that we've presented before. So we have cluster 1, cluster 2. Once we found this cluster, we want to make sure that these clusters are actually clinically relevant. They really tell us something about the patients that can be useful for clinical decisions later. So to check for this, we've compared the delta power for the participant in the cluster 1 to healthy participants, doing the same comparison between cluster 2 and healthy participants. And what we see is that the patients in cluster 1, with respect to their delta power score, they are significantly different from healthy participants. There is really something happening in these patients that's different. But the patients in cluster 2 are not significantly different from healthy participants. And if we compare directly the 2 clusters together, there's a big difference between the 2 clusters. So this really tells us that these clusters are clinically relevant, and we can use that information to inform clinical decisions. And what's not presented here, but what we know right now, and I can say, is that we also have right now preliminary evidence that this relationship that we found here with this biotyping analysis is also associated with changes of symptoms after treatment. So that was this first analysis to show that our pipeline works with one EEG modality, which was power. Of course, we want to make sure that this pipeline works with different kinds of data, that it doesn't really work for one specific case and one specific measure. So that's step 2 of our pipeline validation, where we run kind of the same pipeline on a different, what we call EEG modality, so a different measure from the EEG signal. Previously, we were looking at power. Now we're looking at complexity measure. So complexity basically measures the regularity or how predictable a signal is through time. And so we're measuring that all over the head with different sensors we have over the head of the participant. And why we are doing this analysis is that there is new evidence that brain-signal complexity could be a marker of antidepressant treatment response, meaning that if we measure this complexity, we can tell pretty quickly if a patient is responding to the antidepressant treatment or not, which is a very powerful thing to be able to do. And this analysis also illustrates what kind of research questions we can address with this biotyping pipeline. In the study before, we looked at how baseline biology related to symptoms, but here we can also investigate how changes in biology relate to changes, specific symptoms following a treatment. So this is providing new insight into how treatment may work. So that's also something we can study with the biotyping. So that's work I'm currently doing. And I show here, again, the results from the CCA step that we have on this data. We see, again, this kind of significant correlation that we found between our 2 domains. One is the complexity measure at baseline, and the other one is the change in individual depressive symptoms after 8 weeks of treatment. And as the same as we did with the previous analysis, we can look at which symptoms contribute maximally to this relationship. So we found symptoms such as involvement, irritability, fatigability, hypersomnia. Then we could also see where this kind of relationship is observed onto the topography on the head of the participants. So we can see here that this is contributed maximally by signal on like frontal temporal regions bilaterally and some more like parietal electrodes, central parietal electrodes. So I won't go into too much detail here. So right after I'm done with this presentation, I'll also show you a short video by our colleague Raj, who will demonstrate to you how to get this kind of results using our toolbox. So that was, again, step 2 of validation, using a different measure for EEG. And then that's work we're going to be doing in the next few weeks, 2 months. We want to do step 3 of pipeline validation because we're not happy yet with how robust, like making sure that our pipeline is really robust. We really want to show that this pipeline also works with a completely new imaging modality. So before we're looking at EEG signal, we want to look at a completely different way of imaging the brain activity. So that's work we'll be doing. And for this, we'll use the POND dataset from the POND network. Again, this is a dataset that's available through OBI, and we've been able to have access to these datasets through the collaboration with the Center for Analytics. We're very, very grateful for this. The POND dataset has a lot of brain imaging techniques. We've chosen to use to look at magnetoencephalography, or MEG, which is a technique that looks at the magnetic field from the brain, again, through time. So it's another way of imaging brain activities through time and looking also at clinical scores. And what's really interesting about this data set as well is it's a very different patient group. This is recording from children with neurodevelopmental disorders. What's really powerful about this as well is that this brings together different diagnoses. So I've illustrated here what we mean by neurodevelopmental disorders. So this includes ADHD, autism spectrum disorder, obsessive compulsive disorder, and a few other diagnoses. So this really enables us to see like what biotyping can do across different diagnosis groups. And so our goal here is to identify brain activity patterns linked to depression symptoms across diagnostic groups, but also mainly to show that our pipeline that we've implemented in our toolbox is also robust for a completely new imaging modality. And we can even think of a step forward to validate this pipeline, which would be using still other brain imaging modality that we have available around. So we're really working hard to validate this pipeline. So I'll play now a video recorded by my colleague Raj, who will show you how to do, because I've presented all this beautiful pipeline for biotyping, and I'm sure you're all very curious to see how it works directly within the toolbox. So I'll play this video to show it to you guys.
My name is Raj, and I will provide an example of using the IM Biotype toolbox. In this example, we are looking at a pipeline which involves PCA and CCA for the data fusion step, and an
sorry, I'm trying to increase the volume.
Exploratory cluster analysis. The data sets that we're looking at right now are baseline EEG, looking specifically at complexity metrics, and also changes in clinical scores over the course of a treatment. In the IM Biotype toolbox, you can add datasets and remove datasets, and these can just be raw CSV file formats. When you go to pre-process these, you can start to merge these datasets together using the pre-process dataset functionality, including join. You can filter by rows, you can impute missing values, select specific columns to keep, create formula columns, calculate differences between the rows, such as changes from week to week in terms of clinical scores, and standardization or normalizing. Once you've combined all your data together in suitable feature vectors, you can actually assign those feature vectors to your study. In this case, we have raw feature vectors, which are assigned here, including MSE features, which are the multi-scale entropy or complexity features. We have the quiz features, which are the clinical scale. And we also have covariates for both. Currently, the finalized dataset was the MSE quiz join summary clean. From this dataset, you can go ahead and group your channels, such as the course timescales, and simply assign it here to transfer to your study. The next step is data fusion. You can load settings from a past configuration. I've already created one using our scripting language, so you can go back and forth between the scripting language and the toolbox. Here you can assign your X and Y data features for the data fusion and your CCA type. In Paul's initial study, we use a regularized CCA, and in Chloe's study, we use a classic CCA, but using some enhanced pre-processing for features. For these pre-processing step, you can go in and customize it for both the MSE features and the quiz features. You can enable covariate residualization, for example, and standardization, and PCA is what we used in this pipeline. You can preview all of those here by looking at the feature processing preview and look at all the parameters and steps. Finally, you can assign some statistical testing to your dataset and the CCA run. Once that's done, IM Biotype will generate a preview of the data processing flow, and you can use this to verify that everything looks correct and simply run the configuration. I've run a past configuration, which allows you to now look at some of the results. So as you can see, we have some interesting results that mirror what Chloe presented with the first significant component. We have a second component as well, and we can see all the different components here and their loadings, as well as the topographic view. You can look at parameters as well that was used for the run, and you can also look at the canonical correlations. We're working to implement more robust statistical features into the toolbox display, but those are all also currently calculated in the backend. Now let's look at clustering. So we've been able to do some CCA related work here and fuse the data features together. And we can now do an exploratory analysis just looking at those joint feature vectors. And we could also in the future look at the original feature vectors as well. We can, in the IMBiotype toolbox, change the cluster settings method. So we can look at hierarchical or DB-scan methods, which enable us to identify outliers, or just go with the simple k-means and 2 clusters. You can preview the clustering configuration here, similarly to the data fusion. And finally, we can go and just look at the results of the clustering. And as you can see, we have a very exploratory analysis, but we can see some interesting patterns here in the PC plot. Final step that we will be implementing is the evaluation, where we can actually look at different configurations, processing and clustering, and compare metrics. We can also look at robustness of these biotypes overall and uncertainty estimation for further data sets. Thanks for listening.
Okay, so that was the demonstration in the toolbox, and Raj is also present today for this webinar to answer any questions you may have. And I think that's all for us. Thank you very much for your attention, and we are very much looking forward to answering all your questions.
That's fantastic. Thank you, Chloe. Thank you, Raj, for the video. Very exciting work. So we'll, this is good timing. We have about 15 minutes to have a question and answer period. So if anybody in the audience has a question, feel free to type it in the chat. We can also enable unmuting if you prefer to speak. Please raise your hand if you have a question. That'll be easier for us to go through them. I might start. I know I've taken a few notes here. This is exciting work, and then we've had a chance to touch base over the last few months. But looking at this again, I had maybe some questions that might be interesting for the group as well. First, I was curious about the domains. Chloe, you explained how, and Raj in the demo, you explained how you can load different files and you can look at these 2 different domains, for example. I think it was a really terrific sort of illustration that you had, Chloe. Can you explain how would you work potentially with a 3rd domain or 4th domain, and how does that impact the analysis, and what are the challenges related to adding more domains?
That's an excellent question. So I can start answering, and if anyone from the group also wants to jump in and add some stuff. So I've presented already, so I've given an overview of the CCA method we're using for data fusion. So that's the classical approach. We also have a few variants already integrated within the toolbox that I think Raj presented as like regularized CCA, sparse CCA, which allows like to have more features and kind of like have some kind of penalties for the CCA for this. But if we want to have more domains, because that right now the CCA method that we have and the variance that we have is limited to 2 domains, as you said. But there are some CCA methods that allow, I think, yeah, up to any kind of domains that you want, so 3 or 4. So that's a variant of the CCA we don't have in the toolbox right now, but that's definitely something we're planning on like adding in the toolbox later on. So yeah, it's been a lot of work to get this pipeline working right now, but what we want to do with this toolbox is to continue working on it and adding different variants, different methods that people can use.
That's great. Yeah, and a little bit of a follow-up on that is I mean, you mentioned how the clustering essentially helps you start to subtype and biotype. I think is how you define it, which is really exciting. I was wondering, what if you already had information about like some of these clusters or groups, the participants within your data set, how they respond to a specific treatment, and how could you potentially integrate that knowledge? If you know that they're responding really well to a treatment, would they be belonging to that cluster 1 or cluster 2, and would you be able to visualize that, or how would you go about maybe adding that information? Is it a new domain, or is it something you do sort of after the clustering?
That's an excellent question as well. So I don't know, I think for the visualization, if you just want to visualize where those different types of patients are within our data, we don't, again, it's not a feature we have right now in the toolbox, but it's definitely something we can be open to adding. I think probably Raj can speak to that, because I think he also has some plans of like adding different visualization. As far as using this as a different domain, I'm not completely an expert in this, so maybe I'll pass it on to some of my team members that might have a better knowledge of like the different methods we can use to integrate this information to do the like data fusion or before the clustering, because that's not my personal domain of expertise. So yeah, I don't know if Raj wants to jump in or has any knowledge about this.
Yeah, one of the areas that we're looking into, especially when it comes to the evaluation, is like integrating known information and doing uncertainty estimation. So I think one of the other milestones we're working on is looking at how we can do also the like a supervised framework and a test train, and looking at how it would work overall across multiple data sets. So I think that's something where we can start integrating known information or other modalities into that framework as well. I'm not sure if that answers your question.
Yeah, that's great. Yeah, I was thinking about that. That was actually kind of a third follow-up. So how do you integrate potentially, yeah, supervised, like known outcomes, I guess, which is exactly that, I guess, the supervised approach. So, but this is, yeah, super exciting. I think it's a beautiful outputs as well in terms of like the charting and the graphics. Um, that's great. I don't see any other questions yet, so I might follow up with another one. Um, I was curious about, well, I mean, the clustering itself can be, in my experience, fairly compute intensive. Um, but I guess like with the process of feature engineering and sort of that feature selection, dimensionality reduction, it probably helps quite a bit in terms of reducing that compute. But have you tested different methods that are more compute intensive than others when it comes to the clustering?
I think probably Raj has more experience working directly with the toolbox with the clustering, so I can maybe let him answer. I think for the k-means that I've used mostly, it was very easy to run within the toolbox. But yeah, I haven't tried the other methods myself, so I might let Raj answer this one as well.
Yeah, it usually scales up with the number of participants in your data set. So far, the processing times have been super quick in terms of all the steps, but as we grow, hopefully, number of data sets, number of individuals, then that clustering can get more intensive. And then also some of the visualization. I mean, there's the nonlinear ways to look at clustering as well, which could be more intensive in the future.
Fantastic. I also had a bit of more of a scientific question because you showed some really interesting results there with the delta being correlated with self-reporting on depression. Delta, as far as I know, is in that sort of very low frequency band of, I think, half a hertz, like 0.5 hertz to 4 hertz, roughly. Um, do you guys have a hypothesis as to what might be going on there? Is it some sort of like carrying wave in the brain, or is there like some sort of, are there some ideas, I guess, scientifically as to what the correlation might be with mental health?
I don't know if I should like, answer this question or if like Faranak and Paul wants to speak to their own result as well. But it's been known that the delta oscillation is modulated through depression. There's been a lot of literature on this before. So yeah, I don't know if Paul here or Faranak want to give some detail because I know they've been more involved within the writing and discussion of these results.
Thank you, Chloe. I let Paul speak to it. This is one of his babies. Yeah, and then I can jump into it as well later.
Well, thanks for the great question, Francis. We kind of think of delta power, we frame it more to the parts of cognition. So some of the results we found that Chloe showed included suicidal ideation, low mood, along with some sleep-related items. What's interesting is when we look back to the cognitive neuroscience literature, delta power is very strongly linked to things like inhibitory control, response inhibition, and we know that's very tightly linked to things like suicidal ideation. So one hypothesis is we're capturing some of these more cognitive domains just when the person's at rest. Delta power is very much still being studied as to the origins of it, but that's one working hypothesis we're leaning towards.
Yeah, very nice. So around cognitive control, within Kanban, we look at various treatments, cognitive behavioral therapy being one of them. Some of our other group members have also shown how delta within the context of a task like cognitive control could also be predictive of response to first-line treatments, for instance, in depression. So more work to be done. So the data that Chloe showed are around resting EEG, but you can also look at task and try to start answering some of these underlying networks that are involved as well. There's a hand up by Mahmoud.
Oh yeah, I just have a small clarifying question, but first of all, thank you very much for the presentation. This is very interesting and fascinating work. My question is regarding the signal complexity. Is that the same thing as entropy, or it's different?
That's an excellent question. So yeah, I use the term complexity, but what you can see, if I can show the slide maybe right now, like the measure that we're using is what we call MSE. And so MSE stands for multi-scale entropy. So you're very much right that this is a measure of entropy. There's a lot of different ways of measuring entropy. So there's some that are more like adapted to different kinds of signals. So that's a measure of entropy that's been validated for biological signals like EEG. Because I know there's like certain types of entropy that you measure, like if you have a binary signal, 1, 0, this is not the case for a brain signal, which is like change of amplitude of a signal over time that's more continuous. And so what multiscale entropy as well does, so what it basically does, it measures what we call sample entropy. And so sample entropy, you find different patterns in the data and you count how many times they repeat. And with this, you can know if the signal is more or less predictable. So if it's less predictable, it has a higher entropy. So it might be very counterintuitive for some. But that's the measure, and what we call it multiscale. I haven't gone into detail here, but you can see here, this is a measure of multiscale entropy, complexity, what we call it scales 35 to 70, so that coarse temporal scales. So you can have this measure entropy for different temporal scales of the signal, for a signal that moving faster or slower. So that's also one of the powerful characteristics of this measure of multiscale entropy. So there, yeah, thank you for your question.
Thank you.
Fantastic. Thank you, Mahmoud, for the question. I don't see any other questions at the moment, but we are getting close to the end of the hour. So I first want to really thank you very much, Chloe, Raj, Faranak, and Paul as well for this excellent presentation. Very exciting work. You've accomplished a lot over the year, and we're not done yet. There's still a number of months to go, but it's been really exciting to see the progress of this project and this I think tool that will very likely benefit the broader community in a substantial way. So we're really excited by this. So thank you very much. I want to thank everybody for joining the meeting today. I did want to highlight the upcoming webinars in the chat. So I'll post a link to Dr. Clement Ma's lab work and we're going to have a webinar coming up on November the 11th, as well as another webinar by Ophthalmo, a startup, with Dr. Ihsan Amjadian, leading work on developing foundational models for optometry and helping optometrists with their diagnosis and processing of data in the clinic. So some exciting webinars. So stay tuned for those. You can register on the Eventbrite links that I just shared. But otherwise, I want to thank everybody for joining today. This is recorded, and so we will be preparing the recording to publish on our YouTube channel in coming weeks. So those will also be available. So again, thank you, Chloe. Thank you, Raj. Thank you, Faranak and Paul. We had a great session, and stay tuned for the next webinar.